Barbara, what do you think is the single key challenge in the neuroscience drug discovery research at the moment?
So thank you for the question. I believe that one of the key challenges in the drug discovery research, and in particular in the neuroscience field at the moment, is the lack of robust disease-relevant preclinical models. I believe that, in fact, the poor clinical translation can largely be attributed to the failure of this cellular model. To recapitulate the key pathophysiological features of the human disease, including the complex heterogeneity of the tissue, the intra and intercellular communication and interactions, and also the I would say the penetration, the poor drug penetration through the tissue, and all of which may ultimately have a profound effect on the therapeutic response in vivo. Despite the tremendous progress that has been made recently with the implementation of primary and patient-derived cellular model, 3D systems, organoids, induced pluripotent stem cells cellular model for ex vivo and also in vitro translational research applications. We still, and this is done to overcome, of course, many of the disadvantages of using transformed cell lines for drug discovery. I feel like these models still lack the robustness, the flexibility, and the scalability that is required in pharma research. And this, in fact, has hampered the adoption of these in vitro cell models at the early stages of the drug development process in neuroscience. Therefore, my belief is that although important progress has been made in developing this more physiologically relevant and I would say a predictive cell-based assay in neuroscience, many key aspects of this technology needs to be improved to promote adoption and with the ultimate aim to reduce clinical attrition rates.
Please can you share with us how phenotypic-based and on-target discovery play a part in the identification of targets in neuroscience?
Yes, I believe that both approaches, the phenotypic drug discovery and the target-based drug discovery approaches should be seen as complementary tools that together can increase the likelihood of discovering new drugs with a new efficacious molecular mechanism of action in neuroscience. I would say that obviously the target-based drug discovery plays an important role in identifying drugs for well-validated targets, which means molecular targets that are hypothesized to have a real key role in the disease. Phenotypic drug discovery instead is target agnostic, so there's no need to know the target that drive the disease, and actually offers a mean of recapitulating disease modeling in vitro using complex cellular systems. I would say the unique promise of phenotypic drug discovery is the ability to exploit a disease phenotype to discover real new novel treatment for disease for which the underlying cause is unknown. And this is very well the case for complex and multifactorial diseases such as neurodegenerative diseases, which are largely untractable, not tractable right now. And of course, the target-based approach does not require a cellular system in the beginning, whether the phenotypic drug discovery, based discovery approach require a disease-relevant mechanism of action, which is well understood, and a very, I would say, screening disease model that where a chain of translatability is sufficiently well established. I would say that advances in iPSC models, CRISPR genome editing, and imaging technologies have enabled the development right now of novel cell-based disease models that can recapitulate complex disease biology. And this is actually a plus right now to implement more phenotypic drug discovery in neuroscience. And this is mostly going to be having an impact in rare diseases for which the molecular target might not be known, but the underlying molecular pathway is actually very well understood. And there are several advantages of phenotypic drug discovery in neuroscience and in also other fields. For example, it's possible to develop first-in-class medicines. It's possible to develop to discover novel targets, and we can actually apply this approach in disease for which obviously targets are unknown, but also for which targets are undruggable. One of the key advantages is probably the more translatability in vivo, and also a possibility of discover some side effect of compounds early on, such as, for example, toxicity.
What would you be covering at your presentation?
At the meeting, I will be very happy to present our work on the development of a physiologically relevant and easy scalable Lume cell-based model of mutant G2019S LRRK2 driven Parkinson disease. This is a translational Parkinson disease model, which has been established in Land human mesencephalic cells, or Lume cells, which are actually able to be differentiated in dopaminergic neurons, and has been developed by stably overexpressing wild type and mutant G2019S LRRK2 proteins. We have observed that the expression of the mutant LRRK2 protein causes the rapid onset of different Parkinson disease relevant phenotypes, such as disruption of nuclear morphology, mitochondrial morphology, and also lysosomal morphology. And although our model is not a perfect model for Parkinson disease, because it's still a 2D model, very simplistic, we believe that it still can be used to interrogate LRRK2 biology and to identify novel LRRK2 mediated pathogenic mechanism and molecular targets for Parkinson disease intervention. In addition, this model is very valuable because it can be used in high throughput or high content phenotypic screening and could greatly facilitate the discovery of a novel target or a novel pathway for disease intervention.
What are you looking to achieve at the event?
I'm really looking forward to my participation to this event. I'm really looking forward to expose the findings, so our internal research efforts, to learn about the latest development and the new advances. Sorry, can I start again? I apologize.
No problem at all, please go ahead.
I just restart. Okay. So I'm really looking forward to my participation to this event, to expose our internal research efforts to this meeting, to learn about the latest development and also advances in the drug discovery field, and to establish obviously new contacts try to set up new collaborations and also expand my networks, which I find very important for improving our research.
Gregory, what do you think is the single key challenge in the drug discovery research at the moment?
Yeah, so thank you very much for the question. So I think the biggest challenge in drug discovery at the moment is kind of the same that's in translational research generally, and it's twofold. It's our ability to screen and test the activity of preclinical assets, whether it be for drug discovery or downstream research, in a more physiologically complex and physiologically relevant model system that better models what the in vivo system is going to look like, so where that patient base is going to look like. And then hand in hand with that, a tool chain or tools to robustly quantify and extract meaningful data from those systems. So if we take a look, for instance, at the anti-cancer drug discovery field that we're in, if we're looking at small molecule drugs or IO drugs, the discovery here relies on a lot of cytotoxic model systems, the cytotoxicity of cell line model systems or co-culture models, and if we speak of IO drugs. But these have little disease relevance, and this increases the risk of running in the wrong direction, especially if we look at the fail rate of drug approvals in this space. But if we can start in a more relevant system, and the way that we do things at Allcyte is we work in unmanipulated primary patient material, it provides better foresight for drug action. And this model contains all the necessary cells and untransformed cells and immune cells or cancer cells and immune cells that are present in each patient. So more patient-centric research, translational research, and patient-centric discovery. It provides not only a snapshot of the disease state, but also a snapshot of the diversity of patients that drug may see in a clinical trial. And when we get back to the robust technology, we can't use a plate reader or well-level averaged results, because ultimately that target population of cells, the cancer cells, is a minority within many of these samples. So we leverage high throughput confocal microscopy, single cell image analysis, or deep learning based image analysis, to really extract meaningful data at the single cell level. So what is my drug of interest, or what are my drugs of interest doing in these primary patient materials at that single cell level in each one of these images? And so when we look at the model system, the tech, we believe that future upstream discovery and downstream translational research will rely on primary material. And the first time a drug candidate sees a patient sample shouldn't be in the first clinical trial, it should be upstream, therefore kind of solving these key challenges in model development and asset development.
What would you be covering at your presentation?
I'll be speaking about the deployment of primary model systems and the use of high throughput, high content microscopy and AI or deep learning supported image analysis techniques, along with cloud computing, as key drivers for translational research and target discovery. So the image analysis pipelines that we utilize are custom made, so I'll talk about these, as is the deep learning network architecture we use for cell detection and classification in our images. The images of primary samples are extraordinarily unique in the sense that they contain many different types of lineage cells, big cells, small cells, cells that are in microaggregates overlapping with each other. And we need very robust tools in order to extract meaningful data from all of these images. So I'll discuss this. Primary solid tumor models that enable both target discovery from large library screening and also 3D microscopy and 3D microscopy image analysis, or 3D image analysis, to enable how we visualize multicellular aggregates from solid tumors and how drugs affect the cells within these microaggregates. And I'll talk a little bit also about unsupervised learning and the use of unsupervised learning and cell features of how this can be leveraged for drug activity assessments.
How are the AI approaches of the analysis of high-dimensional profiling data create opportunities to improve many steps in your drug discovery research?
So we leverage AI, in particular deep learning, within our image analysis pipelines. The images we take out of our pipeline are unique in the sense that they contain, as I said, many different types of cells, and each cell has a very unique, let's say, morphological characteristic, especially when you're looking at many different patients, many different cancer types, many different cell types. These cells are big, they're small, they're overlapping with each other. Many of them have diffuse nuclei or more than one nuclei. Many of them have blebbing membranes or they're dying. So for us, we need to have very robust image analysis techniques that can identify and extract meaningful data from every single one of these images. And when we talk about image analysis, we're talking about 13 to 15,000 images in a 3D full plate, and we're talking about 250,000 images a day. So when we make image analysis tools, we're not just talking about speed. I need to make sure that the quality of image analysis, or the quality of data we're pulling out from images of acute myeloid leukemia, are the same quality and robustness of meaningful data that we're extracting from images of patients or images of cells from a patient with pancreatic cancer or lung cancer. And I need to know that everything can be extracted globally in the same way. So the solutions have taken a lot of research and development to perfect, and in fact, we're on our 4th generation of image analysis tool sets that leverage deep learning, the custom network architecture, a lot of training data that has gone into this pan indication, and a lot of technical performance to make sure when we use terms like AI and deep learning, it's not just keywords or buzzwords, it's really meaningful deployment of such tools that extract really reproducible and very detail-oriented data. And we're constantly redeveloping these tool chains to make sure we can extract more and more data from the images that we're getting.
You attended the last drug discovery virtual event. How did you find the event?
So last year I attended the last drug discovery virtual event, and it was actually one of my first virtual events I had attended, and I was happy with the amount of interaction that each guest or each, let's say, visitor or attendee received. A lot of nice feedback on different talks, a lot of Q&A sessions, a lot of roundtable discussions. And this elicited actually a few conversations that led to proof of concept programs from the first conference. And I'm looking forward to the same thing happening here. We get to meet colleagues even during the pandemic and get a really nice sense for how drug discovery is being done within Europe.
What are you looking to achieve at the event?
Allcyte runs on a partnership model where we leverage our tool chains and different image analysis tools, model systems to either look for late-stage asset mechanisms of action or biomarker discovery or upstream drug discovery or target discovery. And so what we're really looking for is the ability to find new partners and find avenues where we can work together with other biotechs or industry or even academic collaboration, so we can continue the design and creation of new drugs and also continue the research and development that we're doing on our image analysis side and model system development. So it's really the opportunity to meet new people we haven't met in the field quite yet, see some interesting talks, and hopefully learn a little bit along the way as well on the next, let's say, stage or generation of drug discovery.
Iolanda, what do you think is the single key challenge in the drug discovery field at the moment?
Well, I think the drug discovery is a long and complex process, as we know. And if we look at the whole process from the top, we might identify more than one challenging step. But let's try to focus on the early stages of the drug discovery, so those which are involved in lead generation. I think the key challenge right now when it comes to lead generation is really how to get the best out of this integrated drug discovery platform that we have. You know, the drug discovery toolbox is a very dynamic box. It is in continuous evolution and it's in continuous expansion. So right now we have many different approaches when it comes to lead generation, and this is really, I mean, it's a luxury. It's really nice to have more than one approach to use, but on the other hand, sometimes having more than one option to choose from, it can also be demanding. Also because all those approaches, they have their own strengths and their own limitations. And obviously, one should kind of take that in consideration when defining the best strategy for the program. If you think that, for example, hydrophore screening, DNA-encoded libraries, fragment-based approach, they're all very successful approaches. They proved to be successful. They proved to be capable of providing tractable hits. However, we do know that sometimes they are sometimes one is successful and the other one, we fail with one and we succeed with another one. And eventually with the next project would be the other way around. So it is really crucial to make the right choice. Also, I mean, as I said, all those approaches, they have their own strengths and their own limitations, but they are also in continuous evolution. I mean, we keep learning from them, we keep optimizing them. So we should also constantly kind of be updated on what is the status of the limitations and where is the status of the strengths for the different approaches. Let me do an example. DNA-encoded libraries. I mean, we right now, I think the main challenge is still how to find tractable hits and how to prioritize them. And the best way to solve this problem is, or to address this challenge, is really to minimize the attrition rate and the false positive rate. And because that's how we can increase our chance to be successful, right? And if you want also the focus of my talk, the DNA-encoded libraries screening in a cell is somehow reconnected to that. I mean, we were not able to screen DNA-encoded libraries inside the cell until now, but now we know that that is possible, and hopefully it will represent a new way to reduce the attrition rate, to reduce the false positive rate, and therefore to increase our success. So I will say that in general, it's important to keep in mind the importance of integration in the early drug discovery.
What would you be covering at your presentation?
My talk will present the first successful screening of DNA-encoded libraries inside a living cell. It's a very important yet exciting achievement. If you think that we are screening 400 million compounds in a single cell, I mean, this is impressive. Conceptually, it's impressive, right? But it is not only a matter of numbers, of course. That screening inside a cell, it also includes other important benefits. For example, we don't need a purified target protein. And that basically means that we are able to access a broader target space, which is really important. In addition to that, we put in the protein in a cell, not in a tube anymore. And obviously this can only be better for the protein. I mean, we are offering a more physiologically relevant environment to the protein. So the expectation is that we will have a lower attrition rate. So it is really important. So I will talk a little bit about that. I will try to touch the most important aspect of the technology. I will show some case studies, and I will also try to talk a little bit of what kind of challenges we've met, because, you know, when it comes to a screening of DEL inside the cell, you will meet some important technological challenges. For example, one is the delivery of the DEL inside the cell. Or another important aspect is how to discriminate between library members binding to the protein and library members binding to other endogenous proteins. So I will show how we kind of solved these problems during my talk as well. So yes, I will basically talk a little bit of everything around this new approach.
What case studies would you be covering at the event?
So about the case studies, I'm not entirely sure specifically what kind of case studies I will present, to be honest. It will depend on a few factors. And also it depends on what I can disclose by the time of the Congress. Right now we have 3 main case studies that we are able to share and disclose, and they are on Pictorial Alpha, Doc5, and ACS2. But things might change, and I might be able to share more data on those case studies, or to add different case studies. I'm not entirely sure. It really depends on what I can disclose by the time of the Congress.
You attended the last drug discovery virtual event. How did you find the event?
To be honest, I really liked the virtual event last time. I think it was really well done in terms of topics, and I think the most relevant and most important topics were covered on a good level. And I must say also the quality of the presentations was really high. So I was really satisfied in that perspective. I think it was one of the first virtual conferences, and despite that challenge, I think it was really well organized. It also allowed a very easy networking, which is quite important. And and I think also the virtual platform was really easy to use, which is also really important. And you know, conferences are always an important opportunity to meet new people and meet new companies, and you know, get in contact with a new science, because that's the basis of the continuous evolution and continuous inspiration for science. So I was really happy with that, and I was really completely satisfied. And also must say, even sessions like roundtables, which are, we know, pretty dynamic and really interactive, they worked. So yeah, overall a very positive feedback from me, and so I have pretty high expectations for the next one as well.